Good evening, and thank you for joining us as Premier Research Lab sponsors this month's Quantum Research Council Practitioner Webinar. I'm your host, Dr. Gary Piercy. Join me tonight as we embark on a journey through the digestive system. But first, a word from our sponsor.
Do you need hormone support related to performance, strength, and vitality? Testosterone may be the most important single hormone that aging men need, and small quantities of testosterone precursors can play an important role for aging women too. Clinical scientists and the medical community are beginning to realize that the best answer to hormone support may be to provide phytonutrient complexes which promote the body's own natural prohormone precursor production. Premier Testosterone is a botanical-based formula that provides performance, strength, and hormone health. Eurycoma is a special botanical that has been traditionally used to support stamina. This formula features LJ100 Tongkat Ali, which offers a standardized aqueous extract of Eurycoma. It has demonstrated support for sports performance and muscular strength in adult males during intensive strength training programs. It has also been shown to support healthy cortisol levels after endurance exercises. In addition to this science-backed foundation of Eurycoma, this proprietary formula also features two key botanical support blends, Eurybolic blend and Cinnabolic blend. Together, they supply a broad range of biocompatible botanical agents which offers a complex phytochemical profile for additional nutritional support. Performance, strength, vitality. Make life epic and support your hormones naturally with Premier Testosterone. Premier Research Labs remains committed to producing the highest quality dietary supplements and strives to use sophisticated and scientifically accepted methods of quality control and assurance. PRL consistently tests raw materials for pesticides, non-organic materials, heavy metals including arsenic, mercury, cadmium, and lead, residual solvents on extracts, pathogenic microbiology, identity, moisture content, and potency. PRL will continue to evaluate innovative and analytical techniques to ensure the highest standard of quality is obtained so you can achieve your optimal health.
I would like to remind you that the Quantum Summit is coming up November 2nd through the 5th. 2 great courses for one low price. We'll have something special on the Quantum Summit after tonight's presentation, so stay tuned. Our guest speaker tonight is Dr. Dickon Weatherby, founder of bloodchemistryanalysis.com, which provides clinicians with extensive and highly informative reference material, educational seminars, and resources to help them get the most of the diagnostic tests they're already using. Dr. Weatherby is also author and co-author of a number of books in the field of functional diagnosis. Tonight, Dr. Weatherby starts us on a tour of the gastrointestinal tract and biliary system from a functional point of view with a look at the physiology of the gut. Dr. Weatherby, thank you for joining us.
Okay, well, thanks for that introduction. This is Dr. Weatherby. Good to be back with you again, and thanks very much to Premier Research Labs for hosting a 3-part webinar series. The last one I did was on the 10 essential tests in any blood chemistry analysis system. So today I'm going to kind of change gears a little bit, kind of coming back to my roots of the work that I really first started out doing, almost 20 years ago. And that's what I call the primary functional diagnostic realm. And I want to cover 3 important aspects of the GI and the gallbladder, from functional physiology, how we become dysfunctional, and then finally the assessment methodologies of functional diagnosis. So I'm going to give you a quick run through of what each of these webinars are going to include. So today is part 1, and I'm going to cover the functional physiology of the GI and gallbladder system. I'm going to be going over fundamental processes of digestion, absorption, assimilation, utilization, and excretion by focusing in on histology, anatomy, and biochemistry of some of the main players of the GI and biliary system. So this might be a review for some. If you're like me, I always love getting other people's perspectives and hearing about other ideas of how we might go back to what I call the sort of the core fundamentals of functional medicine, which is functional physiology. So I'm going to be looking at the GI and gallbladder system through this functional physiology lens, and it's going to provide a real context for part 2 and part 3. So in next session, part 2, I'm going to go to the dysfunctions of the GI and biliary system. This is really about what happens when all of the wonderful physiological systems I'm going to be covering today start to go wrong. And so we're going to look at not only what happens, but potentially what are some of the causes of these dysfunctions. And then in our final session in November, right after the Quantum Summit, so if any of you are going to be at the Quantum Summit, I'm going to be there. I'm going to be talking about my 4 quadrants of functional diagnosis. So right after that is going to be the third and final session. I'm going to cover the primary functional diagnosis of the GI and gallbladder system. And we're going to look through the lens of functional signs and symptoms analysis, functional and nutritional PE, in-office testing, or what I call in-office terrain analysis, and of course my favorite topic, functional biochemistry analysis. So that should give you a sense of where we're headed. But now let's go right back to one of the things that I said earlier is one of the most important topics, I think, for any practitioner of functional medicine, and that is functional physiology. So the functional physiology of the GI and the biliary system. We know that the gastrointestinal tract plays a large role in health and wellness because it's one of the main areas in our body that communicates so closely with our external environment. And so far, some have gone so far as to say that the GI tract is actually outside of the body. Substances don't get into the body until they have crossed that intestinal lumen or the mucosal absorptive surfaces. So yeah, is the GI system or the inside of our digestive tract, the stomach and the duodenum, is that actually outside of our bodies? It's an interesting thought to think about. The mucosal absorptive surfaces is where the body differentiates between what it wants to keep, absorb and take into circulation, and what must be kept out and excreted. So it's a very, very important lining of our digestive tract that really allows us to be able to take in and also to keep things out. So the main functions of the digestive system are the breakdowns of proteins, fats, and carbohydrates. So those macronutrients and breaking them down into their constituent parts. And then we have the absorption of monosaccharides, di and tripeptides, individual amino acids, fatty acids, vitamins, minerals, water, and other nutrients. And then we have excretion of waste. So the GI tract really provides this barrier to keep out harmful, toxic, or poisonous substances and keeping them out of our general circulation. It's essential to have a working knowledge of the physiological process of digestion absorption in order to begin to understand the dysfunctions associated with the GI and gallbladder. And so that's what we're going to be covering in part 2. So this presentation is really designed to give you that working knowledge. It's broken down into the following sections. So the physiology of digestion, absorption, and excretion. So the physiology of digestion, focusing on the neurological, chemical, mechanical, and hormonal influences at play. So in the mouth, we have mastication, the breaking down mechanically of food. We have salivary juices being excreted, and then we have the act of swallowing. In the stomach, we have the release of HCl and pepsin. In the small intestine, we have bicarbonate and pancreatic enzymes. And in the hepatobiliary or gallbladder system, we have bile formation and secretion. So a rhetorical question to ask yourself is this: where does digestion start? Does it start in the stomach or the mouth, or perhaps does it start in the brain? I personally think that digestion starts in the brain with the incoming signals that enter the CNS from the smell of food, the thought of food, the sight of food, or perhaps even the sound of food being cooked. And this triggers a neuroendocrine cascade that kickstarts the digestive process. So despite really where it starts, it's important to remember that digestion is a top-to-bottom process. Problems at the top, if unresolved, can lead to problems downstream. So many of the functional imbalances we're going to be covering next seminar have a genesis in the digestive system. And so this diagram here is really showing us that it's sort of a north to south process. So in the mouth, we talked a little bit in the earlier slide that it's about mastication and the chemical breakdown of food. So let's go into that in a little bit more detail. So the mouth is the first area of actual digestion. Digestion in the mouth utilizes two main processes: mastication, or the breakdown or the grinding down of food into smaller particles. And then we have chemical breakdown of large macromolecules into smaller molecules through the chemical action of saliva, which has a digestive enzyme called alpha amylase. So this is a digestive carbohydrate digestive enzyme. So it requires an alkaline environment to function, which is why the pH of the saliva really should be between 7 and 7.4. So if any of you are measuring salivary pH, you know how important it is to keep it at that optimum alkaline pH for the action of alpha amylase. So there is some evidence that there is fat digestion starting in the mouth with the secretion of salivary lipase, and I'm sure probably there's also mild protein breakdown as well. So we move from the mouth, we move down into the esophagus. The esophagus is all about swallowing. It's the initial peristaltic motion here. So once the food has been masticated, thoroughly mixed with saliva, we swallow, and the food travels down the esophagus, enters into the stomach through the pyloric sphincter, where the next stage of digestion occurs. So what's the stomach involved with? Here's a picture of the stomach right here. So the stomach is the main area of protein digestion. Protein is best digested in a very acidic environment. So we've gone from an alkaline environment in the saliva. As it travels down into the stomach, we get the release of hydrochloric acid and gastric digestive enzymes such as pepsinogen to enable this process to take place. So HCl and pepsinogen synthesis and secretion are under both neurological and hormonal control, and that's called the enteric nervous system. So I want to take a quick look at that hormonal control, because this is really, really important, because so much of the problems that I think are existing in people's digestive systems, and there's a lot of talk nowadays about the microbiome, I think that the health of the microbiome probably has a genesis in the stomach. So really understanding this hormonal control of gastric secretions. So the hormones that directly stimulate the secretion of specific glands in the stomach include acetylcholine, gastrin, and histamine, which can be thought of as cofactors of acid secretion. It's not a strong stimulator of HCl secretion by itself, but in the presence of either acetylcholine or gastrin, it greatly enhances acid secretion when it binds to H2 receptors on the parietal cell. So this is important to remember when considering that a number of our patients may be on antihistamines or H2 blocking medications such as cimetidine. Neither acetylcholine nor gastrin causes significant secretion of stomach acid when histamine secretion in the stomach is blocked by these drugs. So this next stage is the small intestine. Digestion continues. The food bolus from the stomach enters into the small intestine and the digestive process continues. At this point, the food is sufficiently broken down to begin the absorption process. And I'm going to go through that in a few minutes. So the gallbladder is the next one, and its role is in fat emulsification and digestion. Fat digestion is slightly more complicated than protein or carbohydrate digestion, and this is where the liver gallbladder system comes to play through the production of bile, and I'm going to cover that too. We also have to take a look at what we call the layers of the GI. So I show this slide really to highlight distinct histological areas of the gastrointestinal lining. Harkening back to histology class in our first year of medical school, where we were looking at slides of cross-sections of the lumen of the GI. So the lumen itself has many embedded glands, it has lymphoid tissues, it has ducts that secrete HCl, the mucus, and enzymes. As I mentioned before, the gut has its own nervous system called the enteric nervous system. This begins in the esophagus and extends all the way down to the anus. And the number of neurons in this enteric nervous system is about 100,000, almost equal to the number in the entire spinal cord, which is why some have referred to the GI as almost the second brain. So the enteric nervous system controls GI movement, i.e. the peristaltic movement that happens that kind of propels the contents of the digestive tract further down, and also secretions. So speaking of gut movement, you should notice that there are a number of different muscular layers here. So we've got different striated muscle tissue that allows for the churning or mixing of what is known as the chyme in the gut, as well as the longitudinal peristaltic waves that propel the chyme down the gut. So pretty interesting looking at that. So we've got, longitudinal muscles, and then we've also got different types of muscles that allow for more sort of churning of the chyme. We also should note the heavy vascularization that conveys blood to and from the gut and the liver. So let's talk a little bit about GI terminology. I've thrown some terms around in the last few slides. I think it's important that we are all on the same page. So here are my definitions of digestive terminology. So digestion. Digestion is the breakdown of macronutrients from the food in the GI tract into smaller particles. This occurs through mechanical digestion, through mastication and chewing, and also chemical digestion. Factors affecting optimal digestion include inadequate chewing, eating when stressed, consuming too many liquids with food. And so we're going to actually talk about some of these in part 2 when we start looking at, well, what happens when we are under stress? What happens when we don't chew our food properly? And it sets off this chain reaction of events that happens deep into the gastrointestinal system that have huge ramifications for general health and well-being. Our second one is absorption. Absorption is the ability to absorb food after it has been digested. And I use the term food fairly loosely. What we're actually looking at are the nutrients such as the tripeptides, dipeptides, protein molecules, amino acids, triglycerides, and that type of thing. So these move through the intestinal mucosa. Some factors that are affecting absorption, again, these are things to keep in mind for our next webinar. Poor digestion. If you don't digest properly, you're not breaking down those macronutrients properly, you can't absorb them. Impacted material on the intestinal wall. Mucosal atrophy, where the actual lining of the gastrointestinal mucosa starts to shrink. Stress. Gut flora imbalance, and really lack of nutrients to act as cofactors. So again, that's going to give you a preview of some of the things that we'll be covering in webinar number 2. Transportation and assimilation. After the food is digested and absorbed, is assimilated into the bloodstream and distributed around the body. So some of the factors affecting this include poor digestion and absorption. Remember all the things that are happening above. Detoxification stress. Given that the liver is the site of almost all of the blood traveling in portal circulation from the GI, it is the very first place that it all goes to and has to be filtered and cleansed and goes through detox processes and stuff like that. So detoxification stress can be a very important thing to pay attention to. Lack of nutrients acting as cofactors, pH imbalances, and dehydration. And then our fourth one here is utilization. So this refers to the passage of nutrients from the blood into the cell. Some factors affecting utilization include poor cell membrane health. We're not going to really get a chance to look at membrane health, but it's a very, very important thing to be paying attention to. Excess intra and extracellular toxins can affect utilization. And again, lack of nutrient cofactors as well. So just to kind of give us a sense of some of the external anatomy, let's take a closer look at the stomach. In my opinion, this is where a lot of digestive problems start. So I'm going to go over the physiology of the GI so we get a clear understanding of its function. And when you understand function, it's a lot easier to discuss the evolution and cause of dysfunction. Webinar number 2. The first main area of digestion is the stomach. Some of the anatomical points to make here include the different muscle layers, looking at these different striated and longitudinal muscle layers, longitudinal layers, circular layers, and oblique layers. This basically allows the stomach to sort of act as a kind of slightly like a washing machine that agitates the chyme and mixes it up with the chemical secretions that occur. So there are many folds and rugae. These are the folds and rugae in the gastric mucosa. This increases the surface area. At the top we have a cardiac sphincter, and at the bottom we have what's called the pyloric sphincter. And problems are associated with premature opening of these sphincters, especially the esophageal sphincter itself. Again, let's come back to some histology, going back to early first year of med school. So the functions of the stomach: primary digestion and breakdown of macronutrients. We talked earlier about the thought of food, smell, taste, and food. Actually entering the stomach stimulates the release of the hormone called gastrin. And gastrin is produced by these G cells that sit right at the bottom of the gastric pits here. So hormone-producing G cells, and these secrete gastrin. So what gastrin does is stimulates the production of gastric juices, increases the peristaltic movement of the GI tract, and relaxes the pyloric sphincter. Next, we have HCl. HCl, or hydrochloric acid, produced by parietal cells. These are these guys right here, further up the gastric pit. The pH of the stomach that is produced, the pH of the acid that is produced is about 0.8, incredibly acidic. So the hydrogen ion concentration at this pH is about 3 million times that of arterial blood. So as the acid produced by the parietal cells mixes with other secretions, the pH of the stomach goes to be about 1 to 2.5. So why does the stomach do this? Very important. It plays a very, very important role in human physiology. It provides the correct pH for protein digestion to occur. It also sets up the correct pH for the entire digestive process to work efficiently. So when the content of the stomach enters into the small intestine, it is the acidic pH of the stomach that actually triggers the release of pancreatic secretions to continue the digestive process in the stomach, in the small intestine. So you can imagine if you don't have adequate amounts of pH, something called hypochlorhydria, which I'm going to talk about in the next webinar, we don't get that stimulation happening when the stomach contents enters into the duodenum. HCl plays a very, very important role. Given that this is the very first place that food enters into the body, it plays a role of protecting us from ingested pathogens. These might include viruses, parasites, yeasts, bacteria. So the acidic environment of our stomach effectively sterilizes the food before it has a chance to enter the rest of the digestive tract. Another reason why having a good acidic pH in your stomach is vital. So adequate levels of HCl are essential for the optimum absorption of all minerals, especially calcium and iron and zinc. Last but not least, HCl plays an important role in the absorption of the essential vitamin B12. So parietal cells produce HCl and intrinsic factor. So you can see here, a parietal cell secretes hydrochloric acid and intrinsic factor. Intrinsic factor is an essential part of B12 absorption. We also get secretion and activation of pepsinogen from the chief cells. These chief cells here secrete pepsinogen. So again, chief cells release this pepsinogen to the gastric in the gastric gland. Pepsinogen by itself has no digestive activity, but as soon as it comes in contact with HCl, pepsinogen is immediately converted into a substance called pepsin. Pepsin is a very, very potent proteolytic enzyme. It functions most effectively in a very acidic medium, optimum between 1.8 and 3. And if you have a much more alkaline environment in your stomach, pepsin is pretty much inactive in a pH above 5. And then finally, at the top of the gastric pit, we have mucus cells. If you can imagine, there's a lot of very intense substances being produced here from pepsinogen being converted into pepsin. We have hydrochloric acid. And so the body needs to protect itself. And so we have mucus being secreted from what are called neck cells. So very important mucosal protective function. So the entire surface of the mucosal layer of the stomach is covered in a very thin layer of viscous and alkaline mucus, providing much needed protection from not only the highly acidic environment, but also the direct contact of food itself. Small quantities of other enzymes, such as gastric lipase, gastric amylase, and gelatinase, are also produced. So I love this slide. I think it's really exciting to be able to go back and take a look at how all of this actually works from a physiological perspective. So continuing on our trip here, so we've been sort of spending a lot of time here in the stomach. Now we're moving into the small intestine through the movement of chyme into the duodenum. And what happens here is digestion continues via mechanical and chemical digestion. Mechanical digestion occurs via peristalsis, i.e. that churning kind of washing machine type activity that happens. And chemical digestion occurs through the bile, pancreatic juices, and other intestinal juices. So let's continue our journey through the GI. So after the food has been acted on in the acidic medium of the stomach, it passes through the pyloric sphincter into the small intestine, composed of 3 parts here. We've got the duodenum at the top, then we have the jejunum, and then finally the ileum. And small intestine digestion occurs primarily in the duodenum and the jejunum. So upon passing through the pyloric valve, hormonal messengers enterogastrone and secretin are stimulated by the acidic chyme entering the small intestine. Remember I said how important it was that acidic medium entering in through the pyloric sphincter into the duodenum triggers this cascade that happens in the small intestine. And so that happens through these hormonal messengers, one of the most important of which is secretin. So these hormones inhibit the secretion of gastric juices. So as things flow into the small intestine, it basically, that triggers the release of secretin, then that shuts off gastric juices. And now we have the stimulation of pancreatic juices rich in enzymes and bicarbonate from the exocrine pancreas into the small intestine. So the small intestine is an alkaline environment, and the food bolus or the chyme from the stomach is very, very acidic, so it has to be neutralized. So let's take a look at the pancreatic secretions. So right at the top of the list we have proteolytic or pancreatic enzyme secretions. We have proteolytic enzymes. So pancreatic juices are composed of pancreatic enzymes, and in the proteolytic department we have trypsin and chymotrypsin, which are initially secreted in an inactive form called trypsinogen and chymotrypsinogen. They become activated when they enter the small intestine. They remain inactive in the pancreas to prevent the autodigestion of the pancreas itself. Carbohydrate digesting enzymes such as pancreatic amylase, this breaks down starch, glycogen, and almost all other carbohydrates with the exception of cellulose, and it breaks these things down into di and trisaccharides. And then finally we have fat digestion through pancreatic lipase. This breaks down fats into fatty acids, monoglycerides, cholesterol esterase, and hydrolyzes cholesterol esters, and also phospholipases, which split fatty acids from the phospholipid. So what regulates all this? So we have the regulation of pancreatic secretions. So this is where we get into quite a complex biofeedback system. So we're going to keep track of all of this. So acid chyme enters the duodenum, stimulates the release of secretin from specialized S cells. So what does secretin do? Secretin stimulates the secretion of large quantities of sodium bicarbonate. It slows the emptying of the stomach until the correct alkaline pH is maintained. Secretin stimulates virtually no pancreatic enzyme output. It's all about bicarbonate. Bicarbonate has an essential activity for activating pancreatic enzymes that will only function in an alkaline environment. And then finally, the presence of fat and to some extent protein in the small intestine stimulates the release of a very, very important hormone called cholecystokinin, or CCK. And this is produced from specialized cells. Cholecystokinin, or CCK, passes through the bloodstream to the pancreas, where it causes large quantities of pancreatic digestive enzymes to be released into the small intestines. So the presence of an alkaline environment in the small intestine provided by the bicarbonate activates the pancreatic enzymes, and the process of protein, carbohydrate, and fat digestion continues. Let's take a little side journey here, taking a look at the hepatobiliary system. This has a primary role in fat emulsification and fat digestion with the production of bile. So bile is produced by the liver, and it is stored in the gallbladder. So if you look at the anatomy here, and this is going to be important because we're going to be talking about a test in our final webinar in the assessment section, a test called Murphy's sign. And Murphy's sign is where you actually put some pressure on the liver, and coming through underneath the right rib cage, you can actually start palpating, not the gallbladder itself, but you actually start putting a little bit of pressure on the gallbladder. It's a really good test for monitoring whether or not someone has some gallbladder issues. So bile salts emulsify triglycerides, breaking them down into small chylomicrons so that they can be absorbed. And the rate of bile production is controlled by the autonomic nervous system and hormones produced in the gastric mucosa. So parasympathetic stimulation and CCK. And I just talked about CCK a few minutes ago, but I'll talk about it a little bit more later. So some of the functions of bile, we're going to go through these: fat digestion and emulsification. One of the interesting things to remember is that on an average day, the liver secretes 600 to 1200 milliliters of bile. That's a ton of bile. So bile has two important functions, fat digestion and emulsification. It contains bile acids that act on fats in the intestine. So the first thing it does, bile acids have a sort of a detergent action on fat in the small intestine. And basically this decreases the surface tension of the fat molecules, helps emulsify large fat molecules into tiny globules that can then be further broken down by lipase secreted in the pancreatic juices. So it's a little bit like kind of a bar of soap kind of thing. It helps to break down the surface tension of the fat molecules and helping with their emulsification. Second thing they do, they also aid in the transportation and absorption of fatty acids, monoglycerides, and cholesterol through the intestinal mucosal membrane by forming what are called micelles. So without the action of bile acids, up to about 40% of lipids will be lost in the stool, which is why, when people have problems with bile acid secretion, they oftentimes have fatty stools. So this is a major problem for the millions of people that unfortunately have had their gallbladders removed. We have this constant drip of bile that's not that concentrated, so we don't get this thing happening. And so about 40 to 50% of lipids which could be absorbed are not, and they simply pass through the stool, and you can actually see them on the other end. Bile plays a major excretory function by facilitating the excretion of the following: bilirubin, the end product of red blood cell or hemoglobin destruction. So it's also an excretory process. Excretes excess cholesterol and is probably one of the most important tools the body has for getting rid of fat-soluble toxins. So what about the regulation of bile secretion? This occurs in two distinct phases. So bile is formed initially by liver hepatocytes and contains large amounts of cholesterol, bile acids, phospholipases, and other constituents. The bile formed collects in the tiny bile canaliculi. Again, if you can remember back to the histology, we've got these functional units of the liver, and you can see these tiny little bile canaliculi. If you can remember back to the slides themselves, like little holes where the tiny little bile canaliculi, where the bile flows collecting into progressively larger and larger tubules, finally coming into the terminal bile ducts before reaching the hepatic duct and the common bile duct. And from there it can go into two directions. One is directly into the duodenum or through the cystic duct into the gallbladder. So the gallbladder forms the storage place for bile. It stores bile produced by the liver. It can only store a relatively small amount, between 20 to 60 milliliters, but the gallbladder has the capacity to absorb electrolytes and water, thus concentrating the bile salts, lecithin, cholesterol, and bilirubin. So I talked about earlier about 600 to 1200 milliliters of bile. What ends up happening in the gallbladder is it gets progressively more and more concentrated, and therefore we have much more effective emulsification when it's excreted into the duodenum. On the other hand, we also have a problem whereby people can also get sort of crystals forming in gallstones, and we'll talk more about that in webinar number 2. I promised I'd talk about CCK. So this is a very, very important hormone. I'll talk about it first in terms of fat digestion. So amino acids and fatty acids in the chyme in the small intestine stimulate the release of CCK. So CCK does a number of different things. It inhibits gastric emptying, stimulates the secretion of pancreatic juices. It induces a feeling of satiety. So it's one of the main things that tells you, I'm full, I don't need to eat anymore. And finally, it causes the gallbladder to contract, thus ejecting bile into the small intestine. And this is the part that I want to pay attention to here, because this has a role, I think, in the development of gallbladder issues. So the most potent stimulator for gallbladder emptying is the hormone CCK. Causes rhythmic contractions of the muscular walls of the gallbladder. So similar hormone that causes an increase in secretions of pancreatic enzymes, and in the case of the gallbladder releases the stimulus for fatty acids in the duodenum. Cholecystokinin stimulates the emptying of the gallbladder, causes two things to happen. Causes the relaxation of the sphincter of Oddi. Remember back to our anatomy, the sphincter of Oddi. The sphincter is at the junction of the duodenum and the common bile duct, and we get rhythmic contraction of the muscular walls of the gallbladder. So it's clear that there are other neurological systems that are at play in the control and regulation of digestion. The enteric nervous system appears to be one of the main agents for close regulation of these systems, and the enteric nervous system utilizes a number of neurotransmitters to conduct this function. But one of the biggest players in the release of secretions of pancreatic juices and also the release of bile from the gallbladder is the hormone CCK. Let's take a look now at absorption. So we've digested, we've emulsified, and now we want to absorb. So this can occur pretty much anywhere throughout the GI. The stomach has a poor absorptive surface because it lacks villi. The villi of the small intestine that increases the absorptive surface area. But certain lipid-soluble substances such as alcohol or aspirin can be absorbed in small amounts through the stomach lining. But we're going to pay attention mostly to the small intestine and its role in absorption. So we discussed the small intestine's role in digestion. Now let's take a look at it in terms of absorption. So the absorption of nutrients occurs primarily in the small intestine, and success depends on a lot of the activity that happened upstream with the stomach, the gallbladder, and the pancreas. All of these have to be functioning optimally in order for adequate absorption to take place. So one of the first things to consider is also that absorption of nutrients requires a large surface area. And the small intestine has evolved to be the main absorptive organ in the body, and it does this in 3 different ways. One is through the concentric folds of the lining of the small intestine. That's what this particular micrograph is looking at. Micrograph is looking at these tiny folds of the small intestine. And this increases the surface area by about 3 times. Then we also have finger-like projections called villi that project out into the lumen of the intestine. And that's a sort of a cross-section of a villi here. And that increases the surface area about 30 times. And then each epithelial cell in the small intestinal lumen is covered in hundreds of microvilli that increase the total surface area by 600 times. So the various nutrients, fats, vitamins, amino acids, monosaccharides, minerals, and water are absorbed via a passive, facilitated, or active transport system. Should be noted here that passive, facilitated, and active transport takes place through the cell itself. This should be differentiated from what we call paracellular transport, which is the absorption of substances between the actual intestinal cells through what we call the tight junctions. So the tight junctions are what allows water and electrolytes, but a loss of integrity of those tight junctions, these are the junctions that exist between the cells themselves, a loss of that integrity of the intestinal lumen caused by inflammation, caused by infection, can cause larger molecules to move through these tight junctions. And I'll talk about that in our second webinar. So this gives us a condition called intestinal hyperpermeability, or in layman's terms, leaky gut syndrome. And like I said, we'll cover that at a future presentation. So what about the physiology of gut flora? A lot of discussion nowadays about the microbiome. I'm not going to be giving a long discussion about the microbiome. Suffice it to say, I'm just going to be talking about some of the main players and the roles that they play, and in talking about the physiology of gut flora. So first of all, the functions of GI flora. Remembering that the GI tract is an ecosystem, we have a balance between aerobic and anaerobic microorganisms known as the microflora or the microbiome. It's been estimated that there are over 500 species of bacteria, and there are many, many more bacterial cells in the intestine than there are in the entire body. The majority of the bacteria are located in the colon, and so in a healthy gut, the dominant flora is composed of beneficial bacteria of the Lactobacillus and Bifidobacteria species, and the microflora are essential to our well-being. So these bacteria provide very real beneficial effects to human health and wellness. So functions of the beneficial bacteria: produce vitamins such as folic acid, vitamin K, and B12. They nourish the lining of the colon by feeding on vegetable fibers and producing butyric acid, an essential fatty acid and a primary fuel for the colonic mucosa. And adequate butyric acid levels reduce the chances of colon cancer. Another function is that they limit the population and growth of harmful bacteria. So we have this dance between beneficial and harmful bacteria. And so the more beneficial ones you have, the less room there is for harmful bacteria. They also break down toxins. They assist in the process of digestion. And so when our gut ecology is in balance, we thrive. And when it's out of balance, we don't thrive. Types of flora: Lactobacillus acidophilus and bifidobacteria. There are half a dozen types of bacteria present in the gut in large numbers. This is changing all the time as more and more research is being done. But Lactobacillus acidophilus in the small gut and bifidobacteria in the large gut are two of these primary strains. Origins of the flora. So just before birth, an infant has a sterile GI tract, although there is some evidence pointing that may or may not be the case. The development of the flora in a newborn depends on whether the child is breastfed or bottle-fed. In breastfed children, the flora of the GI tract is quickly dominated by a beneficial flora called bifidobacteria and a few other types of bacteria, and if left undisturbed, this good flora remains stable until the child is weaned and changed over to solid food. And this sets the place for the development of typical adult flora. In bottle-fed babies, a different microflora develops in early infancy. The population is mainly lactobacilli, with a much lower incidence of bifidobacteria. So there's a large body of evidence to suggest that this difference in the makeup of the flora is connected with the greater susceptibility of bottle-fed infants to infections by harmful microbes that typically cause mild to severe diarrhea. So what are some of the beneficial effects of flora? There's 4 of these. So it provides protection to the GI tract by interfering with the ability of some pathogens to adhere to the surface of the gut lining, thereby limiting the colonization of microbes such as E. coli, Candida, Giardia, and other things. Secondly, it produces natural antibiotic effects. Lactobacilli are believed to produce substances that suppress the growth of potentially harmful organisms. A third role it plays in starving out pathogenic microorganisms, and many harmful microbes require iron to be highly active, and our bodies have a mechanism to minimize microbe growth by ensuring that very little free iron remains in the blood. In the gut, bifidobacteria appear to aid in binding iron, making it unavailable to microbes by concentrating up 1,000 times the normal content. Fourth, increases the efficiency of the body. Bile salts can inhibit harmful flora. By recycling bile salts, the helpful flora maximize the levels of bile in the system. This conservation saves the body precious energy and increases the protection of the gut. So take a look now, we're moving down to the final area of the GI tract, and that's the large intestine. So we're looking here mostly at the role of excretion. By the time the chyme enters into the large intestine, we are done really with our digestion and absorption, although some absorption can happen through the large intestine. Ultimately, it's mostly about recycling and reclaiming water. So a major function of the large intestine is excretion, through the formation of the feces that will eventually be expelled from the body. The large absorptive surface of the colonic mucosa is perfect for the reabsorption of water. So the liquid chyme is very rich in water, hence the fact that it's liquid, and the body wants to recycle that. The body wants to hold on to that water. So the large intestine provides a place for the reabsorption of water and the solidification of the feces itself. And it also provides a place for the storage of feces. It's important to remember that the bile that gets secreted into the small intestine is also an excretory product. Remember I talked about how the gallbladder and the bile was actually one of the most important places for the elimination of fat-soluble toxins. So important that the feces is expelled in a timely manner, because we don't want that reuptake of the bile to happen too much. So if we have constipation or we have slow bowel transit time, this increases the chance that we get a thing called enterohepatic recirculation of toxins, primarily from the bowel. So the longer the feces remains in the body, so if you have a pretty chronic constipation, you're going to get a lot of these toxins is just going to get reuptake back up into the body through the bloodstream and back into the liver. Dysbiotic bacteria can strip toxins from the bile and allow them to be absorbed back into hepatic circulation. Good reason to make sure that the colonic flora is a healthy one. Another great reason for a healthy flora in the colon is the colonic bacterial environment. Produces a substance called butyric acid or butyrate. This is a natural food for the colonic mucosa, produced from available fiber in the colon. The colonic bacteria also produces certain B vitamins, including B1, thiamine, B2, riboflavin, and B12, as well as vitamin K. So that's our journey through the intestines and looking at digestion, absorption, assimilation, and excretion. I know this has probably been a review for many of you, but I hope that it's a good refresher, because I think it's going to provide us with a real opportunity to have a good, meaningful conversation in our next webinar, which is going to be next month, and I'm going to be covering dysfunctions. So we're going to look at what happens when all of these physiological systems that have evolved to happen and function in relationship to one another, what happens when that gets out of balance. So the interconnectedness of the GI systems, the homeostatic balance that exists within the GI, what happens when they get disrupted? What happens in sort of immune issues and allergies and inflammation, and all of that type of stuff. So thanks so much for sticking with me. It's a little shorter than I had imagined it would be, but hopefully you got some good information, and I really look forward to being with you next month, where we talk about the dysfunctions. So this is Dr. Weatherby saying thanks so very much, and I'll be with you next time.
Thanks, Dr. Weatherby, for a review of functional gut physiology. We're also looking forward to Dr. Weatherby's presentation on the quadrants of functional diagnosis at the Quantum Summit coming up November 2nd through the 5th in Austin, Texas, where you can get 2 great courses, 4 days worth of information for one low price. Learn the latest QRA and VASTU information on Thursday and Friday, and then join our great lineup of speakers on Saturday and Sunday for a clinical nutrition course. Speakers like Dr. Caldwell Esselstyn, who'll talk about the effectiveness of dietary intervention on some of the worst chronic conditions. Speakers like Dr. Weatherby, who will discuss the efficacy of functional diagnosis. Dr. Barry Tan, who will cover the latest vitamin E research. Dr. Randolph Aguilera on biocompatible dentistry, and much, much more. Not only will there be great information, but chiropractors can also get 12 hours of CEU credits. And remember, I said there'd be something special about this at the end of the presentation. And here it is. Don't miss out on this great opportunity. We'll be offering a $100 product credit if you register by this Friday. Looking at the rest of the QRA schedule this month, there was a level 1 seminar that is, in Seattle. Tava said that went really well. There'll be another one in Orlando next week, and yes, Orlando is still on, despite the problems with the weather. Also this month, level 2 seminars will be held in Minneapolis, San Francisco, and Secaucus. The QRA schedule is available at quantumresearchcouncil.com, where you can also register, prlabs.com, in the monthly PRL newsletter, or from your account executive. Premier HCL is our webinar special. Buy 6, get 1 free through Friday. We recommend taking Premier HCL after a meal. That way your stomach still maintains production of gastric juices, and we can make sure that the stomach pH is low enough to trigger the rest of the digestive process. The monthly special for September is Premier Testosterone for natural phytonutrient support related to performance, strength, and stamina. Buy 6, get 1 free through the end of the month. Be sure to tune in for our next presentation on October 10th as Premier Research Lab sponsors the QRC Practitioner Webinar Series. Thank you for joining us. I'm Dr. Gary Piercy. Good night.